Okay, I would like to welcome Dr. Reut Abramovich. She is a scientist, space enthusiast, and entrepreneur with a diverse background in academia, research, biotechnology, and space exploration. Welcome, Dr. Abramovich.
Thank you very much. Happy to be here. Thank you for the invite, Nicole.
My pleasure.
So I'm an astrobiologist by training. I'm a fellow international of the Explorers Club. I'm also a visitor lecturer and chair of space sciences for International Space University. And I recently co-founded a biotech company in the area of cement and concrete called Starstone. And I'm also an explorer of extreme environments, which we're going to touch a little bit today when I'm going to talk with you about my vision of what we can do on Mars. And when we have a Martian planetary expedition, what should we look for? So without further ado, I'm going to share my presentation with you, and I hope you will enjoy it. So thank you again for the invitation, and I'm happy to be here. One small note beforehand. There might be an interruption during this thing. I might need to go away for a while for 15 minutes while we're doing this presentation due to an emergency that might occur. Please don't be afraid and don't worry. I will just shut down my Zoom and sort of like I won't log off, but you won't hear or see me, but I'm going to be okay. And I hope I'll be back in about 10 to 15 minutes if everything is fine. So this is just a note beforehand how things are. I'm currently based in Israel, and sometimes we have some issues in the neighborhood.
Thank you
So
for letting us know. Thank you. I appreciate that.
no worries. So let's share. Okay. All right. I need to put it in presentation mode. I remember there was this thing. Right. Um, One moment. Got it. Thank you. So I don't know if you can see this.
Yes, we can see.
Excellent. So basically, as a planetary scientist, I also work in extreme environment. On the right side, it's me in a space simulation mission that occurred in 2018, where I was microbiologist and I was a science officer in a mission that went into a hyper-arid area in the Negev in Israel, in the south of Israel, a desert. And on the other side is something I was able to accomplish a long life dream, and I went to the North Pole to do some research there as well, and the conditions were very, very hard. So the question arises, what do I do and why do I do it, and what relevance it has for Mars? So this talk today is going to be very specific. It's going to be about recent research I did together with my student, Dr. Yael Hellman, and it's an analysis of forward and backward contamination in a Mars analog mission, which is a simulation mission. And it comes from the idea that if you do send a human crew to Mars or to the moon or to other planets in the future, they would go with their own, consortium of microbiota. And the equipment would have also quite a bit of microorganisms on it. And if you are interested in science and you're interested in keeping everybody healthy, you need to find a way in which you can monitor the situation and also understand how much you're carrying, how much bacterial load are you carrying with your equipment and with your human crew when you're going to a new environment like Mars. So astrobiology is a you probably heard this before. I mean, you had some excellent lectures before as well. But basically, astrobiology is a multidisciplinary science. It talks about the origin of life on planet Earth and in other areas as well. How does life evolve? What are the building blocks of life? How did the first DNA molecule look like? How was the first membrane? What kind of atoms and what kind of molecules were involved, for instance, with proteins? We're talking about habitability a lot, and that's where my work usually comes when I talk about extreme environments where the temperatures are extremely high or extremely low. The salinity levels are extremely high, like the Dead Sea here in Israel, which is 375 grams salt per liter. It's highly saline, but we still have life there. We're talking about Mariana Trench. It's 11 kilometers depth. How do life survive in this environment? And in parallel, if they can survive these harsh environments here on Earth, can they survive them elsewhere in other planets? So a lot of my work is also involved with different types of technology and how we can detect life elsewhere on other planets. I do not use telescopes. I do not use James Webb, even though I would love to. But basically, my tool, as many planetary scientists' tools are, is going into the field and making sure that we have the right protocols and the right equipment in place in order to make those observations and find those microorganisms or life forms in places that they're never been found before. When we talk about contamination, we have in our mind all these Hollywood movies about, oh no, we encountered an alien life and they came back to Earth, or they affected the crew and they killed them, or mutate them and stuff like that. But actually, after what I'm going to show you, I think you're going to be much more relaxed. and I will show you how we can basically a proof of concept of research that was published, and how we can detect these kind of things and where the research currently stands. So forward end contamination. Can I move this somewhere? That would be so nice. So forward and backward contamination, what are we talking about? So forward contamination is when we come from planet Earth, and for instance, a very known example in recent years is that Israel sent a Bereshit spaceship that crash landed on the moon, and about half a year later after the crash, it turned out that accidentally it had a payload which included tardigrades, which nobody knew about it beforehand. And that basically is a good example of forward contamination, when we brought something from Earth to an extraterrestrial planetoid or celestial body. Another example, for instance, is the fact that when Apollo astronauts had to dump everything from their ship in order to make the voyage back home, I think it was a Apollo 11, but I'm not sure. But basically what they had to do in order to make that happen, they also dumped a lot of their diapers, which had a lot of course, contamination from their own, biological metabolism. And those diapers with everything in it are still on the moon. That's forward contamination, when you take Earth and you spread it out. Backward contamination is what we see in Hollywood movies, and what we never, ever found anywhere is that when you have something coming back from a space mission and it brings its own endemic population. So we have no known example of that. We haven't detected anything, not when we got back lunar regolith and lunar rocks from the Apollo missions. And at the moment, we're making sure also that return missions from Mars and from other places, when they come back and land on Earth, they're very much double insulated or triple insulated, and we're making sure that there's no contamination from them back to planet Earth. But this is what we talk about contamination. And contamination is a huge, huge issue in space mission planning. There is the I want to say interplanetary, which is of course not, but the planetary for I want to say IPCC, which is the incorrect one. But you have the contamination, you have the Planetary Protection Committee, and that committee is under the UN, one of the UN government authorities, and they have their usually annual conference, not to mention a very long document in which they state how you can avoid contaminating areas on Mars, for instance, and what kind of protocols you need to put in place when you plan where to put the spaceship, what kind of sterilization procedure you can do, bearing in mind that there's no such thing as 100% sterilization method. So you always have to take into account that something from Earth might get onto Mars, and you don't want to contaminate, for instance, another life form. The significance of protection against contamination is ethical, scientific, and safety significant. The safety is very obvious. We want to protect the Earth and the Moon biosphere, for instance, or Mars biosphere against contamination. We don't want to introduce new species. That's one of the reasons why you have also the Antarctic Treaty. We don't want to bring outside stuff, outside life forms into Antarctica. So we have the Antarctic Treaty, which forbids bringing, for instance, dogs and other things since 1967. And also, we have the scientific significance. If I want to say I found life on Mars, I have to be 1000% sure that that life form did not came with me on my equipment, whether it's a microscope or whether it's a rover. You have to be sure that from a scientific significant point of view, the integrity of your data is complete. And we do that when we protect against contamination. And we have the ethical significance, which it goes with the safety significance. Is basically you don't want to interfere with the local life, for instance, on Mars. If there is life forms on Mars, you don't want to interfere with their evolution in some way. So you have to be very careful on how you treat your crew and your equipment and how you spread them around. So we do a lot of analog missions here on Earth. The big space agency does do them as well. NASA, ESA, the Chinese Space Agency, but also you have a lot of citizen science, I would say, organization. For instance, the Mars Society has two big, large areas where they do annual analog missions, for instance, in the Devon Island and also in the Utah Desert. And in these analog missions, also the space agencies, we can do a lot of experiments, we can repeat a lot of experiments, and we can test a hell of a lot, really a lot of equipment and the human-machine interface. And all this is done so the space mission, when it actually occurs, whether it's in the ISS or on the way to Mars, they will work as smoothly as possibly. And all the different problems that can arise, we try to find them out here on Earth. And space mission simulation here on Earth usually mimic one or more conditions that are similar that they're going to encounter in a space. So it could be the isolation, it could be the materials. And so we have many different areas in the world where you can now, even you can go and do space simulations. The Mars Society is one of them. You have a few in Europe, like the Austrian Space Forum and Luna Ares. You have a few in Israel and in the Middle East. And you have, of course, those that go with the space agencies. For instance, here on the bottom, you see something underwater, and that's called Project Nemo. NASA has an underwater facility where astronauts during their training spend time between 2 to 4 weeks and use a lot of the equipment they need to, how to put the suit on, how to operate in a hostile environment, what kind of equipment can work, and it mimics different gravity in a hostile environment. Other places like desert rats, for instance, which you can see up here, there's some with two spacesuits and a vehicle basically looks at this integration between spacesuits that are integrated into the vehicle and how that works in the field in an area that simulates some characteristics, geomorphology, mainly in geology and geographical features that you can find on Mars. So simulation space missions are very important to test different issues, not only technological ones, but also human crew dynamics, psychology, and behavioral issues. So my research together with Yael was we wanted to use really advanced molecular microbiology tools to better understand the cross-contamination potential when you have a human crew on Mars. And we had the perfect place to do it. We had a hyper-arid desert here in Israel. We had a simulation going on for 11 days. Yael was one of the astronauts, the analog astronaut. You'll see her in a few pictures before. And we wanted to test if forward contamination occurs, then we should be able to see changes in the microbial population diversity and its composition. And it would be affected by the introduction of new taxa, new taxonomical groups, new groups of bacteria. Some of them might be human-associated species because the human crew is there. Now, if backward contamination occurs, we would expect to see something coming from the environment, from the soil, coming back into the habitat. The study design included the analog center, which is called DMARS, Desert Mars Analog Hormone Station, situated again in the south of Israel, near Mitzpeh Ramon. And this was an 11-day simulation in 2019. And we had 6 isolated analog astronauts that had performed daily EVAs with simulated spacesuits, weighing about 15 kilos, not so much, but fully suited with a simulated spacesuit, with a helmet, with communication and personal life support system, and they were all living isolated from the rest of us. Also, the Mission Support Center was a 20-minute delay communication between the mission support center and the astronaut themselves, because that mimics very well the distance, excuse me, the communication lag you can get when you have a crew on Mars and a mission support on Earth, because where the planets are and the distance between them takes a very long time to get a communication. So some people would say hi, and until mission support gives you a response back, it could get 40 minutes. That affects the whole way the crew operates on the ground. But coming back to the forward contamination site selection, we chose 3 sites in order to check our thesis, in order to check our theory. And we had 3 sites. So 2 of them were very close to the habitat themselves, and 1 was really far away. It was about 280 meters west of the habitat. It was sort of like marked as a place where only Yael, only the designated analog astronaut can visit. And it wasn't frequent, not by a rover, not by other people, not by the support crew, not by anything. And so we had these 3 places to test. We sampled the first 5 centimeters of the topsoil. And what we did is that we collected it, in the 3rd and 5th and 7th day during EVA. We did DNA extraction within the habitat in a biohood in a very sterile environment as best as we could. And we then later on analyzed those results to see how they fare. Is there any changes towards during the days, and also compared to control. So after the mission, we amplify. So I don't know how much here have a background in molecular biology, but today, what we do today basically is that we can amplify a segment of DNA of any bacteria that we find in the environment. And basically what we do is that we look at the same segment of DNA in different microorganisms. And that the specific segment of DNA is very conserved in evolution, and it gives us a very precise, each DNA segment is slightly different than the other. And we now know how to categorize those differences and so not assume, but after we profile each of these DNA segments, and we can actually characterize which bacteria was found in the sample, how much of it is there, what kind of other characteristic it might have. So basically what we're doing DNA profiling of the soil, of the microorganisms that are there, and we sequence this gene, which is called 16S. So this is Yael. She went out on a designated day and time, and she had to pick these things very carefully because she didn't want to contaminate them with her own gloves when she's picking up. So she had to really, I don't know if any of you have done simulation analog missions, but it's pretty hard sometimes to do these things. You have to be very physically fit because you're carrying a lot of weight. And you have to be very careful when you do microbial work in order not to contaminate it with your own microorganisms that's coming from your skin. And all this situation, to do it within a suit is very, very hard. The helmet does not allow you to hear anything, doesn't necessarily give you the best view that you can have. The gloves she wore here was actually very nice gloves. You can see they're like usually lab gloves, but for other experiments we had, she had very bulky gloves, which make it very hard to work with miniature tools and things like that. So you have to take a lot into consideration what kind of suit you have and what kind of equipment you have in your field when you want to go and do something that, for me as a field microbiologist, is very simple to go into a soil sample. But once you go into space simulation, you have all these astronaut tools and spacesuits and all these different equipment on top of that. This is a short video. Of course, she's going out with someone else who is able to take photo. And just notice how many times she needs to go down on the ground and stand up, down on the ground and stand up. It can be very, very hard. It's really important to have recordings of your scientific activity in the field. So if you want to repeat it, you can make it better. Or if you feel something went wrong, you can have visuals on what went wrong. And of course, it's very important because then you can monitor the other analog astronauts and see if they need assistance, if they need help, if something is wrong. So all these things are together. So she was able to do this repeatedly through the 11 days. And to go, the sites that you're seeing here are the sites which are close to the habitat. as, and you don't hear anything because all their communication are inside the helmets. So you won't be hearing anything at all. There she is. Um, okay. So what we do is once we have all these DNA profiles, we start seeing what are the differences between them, and we start to cluster those which are more similar to one another. So basically what we see here is a what we call a principal coordinate analysis of all soil samples. When you have all, when the samples that are more similar to one another would be clustered closer to one another on this specific, I would say, image of graph or coordinates. And what we're seeing here is basically that we had a very nice, strong separation between the three different sites, which means we found that what we hoped for is that they would have distinct population, each of these areas. And we really hoped that the far side, the one that didn't have any analog astronaut except Yael, would be very different than those that are close to the habitat. And this would be we would be happy to see the differences that are happening occurring today, but this is what we see here. We see a separation between the three different groups. Now, what you see here, let me explain. Each column 3579 is the day of the sample, and the relative abundance, okay, the thickness of the color, the thickness of the steps here, basically of each color, means how much of a specific taxa, of a specific, I would say, family, we find here the relative abundance just according to the number of sequences of that family that was found in the soil sample. So for instance, if we get on the right side under number 7, you see all these grayish, gray-green, I don't know how to call that color. But what you see, that's all related to a dominant family called Formicida, which is basically a cyanobacteria. It's a photosynthetic bacteria. They can, get CO2 and get CO2 and expel O2 and do photosynthetic. That's what cyanobacteria does, and they're tough. Most of them are tough bacteria that are known to be in desert area and in a hyper-arid area with not a lot of water. And what we see, but what we're really interested to see is how many colors we find and what are the changes of the colors when you look through the days. So basically on the left side, you see the far side, which is our sterile side, which is basically what's going to be in the hyper-arid environment of the Ramon crater anyhow. And we do find some dominant families, which we don't find at all in the entrance and in the entrance area. So we find two different, I would say, composition of population, one which is closer to the habitat and one which is further than the habitat. And what we see is we see also a difference in diversion, in the diversity. Basically, what we were looking for is that when you have an extreme environment, you usually don't get a very diverse microbial population because you would find species which are highly adaptable to those specific environment, to that specific temperature, salinity, humidity, UV, and solar radiation. And it won't change a lot because it's an extreme environment. So you'll have a very niche, a very, I would say, like a boutique composition that is really can work with that environment. But when you go to the area where humans come and go in the entrance sites, where you go with your boots in, you go with your boots in and out, then you're sort of like spreading around everything, and those areas are a lot less arid. They're much more they're under shade, and you have a lot of people coming back and forth and spreading them around, and it's quite diverse. We found different species on the sanitary site, which is close to the habitat, and we find a few which were human associated, but which are not pathogenic. It's important to say that. But we did find, of course, much more species which are human associated and are known from other areas where humans sort of like to live. But we didn't find those species in the sterile sites. So we did realize actually that we do have a lot of influence from the human crew around the habitat versus the sterile site. And in the sanitary site actually was the least diverse, and the most prevalent was the Psychobacter, followed by the Actinobacter species. And you can see the changes with the arrow you can see how it changed, how some changes occurred with the time, and it was still the least diverse compared to the other two sites. So when we look at forward contamination, okay, so the sanitary site, why we call it the sanitary, because that's where we had our big, I would say, big sanitary, oh man, I forgot the name. I'm not going to say jerry cans, but basically where we had all the different things related to the restrooms and the toilet area. Basically, the sanitary site represents the highest relative abundance of human-associated genera, and these are the arrows which are going to that. And the seventh day being the most contaminated. So we saw this progression of relative abundance of human-associated genera that just came piling up and getting, much more prevalent as the day went by. And so we were looking at, so that was forward contaminations in the area close to the habitat. But in the sterile site, we didn't notice any contamination as far as we could see. And this is a very high resolution method to check for a contamination. When we looked for backward contamination, so we had to ask ourselves, what do we want to sample? Because if we're saying things are coming back from the outside, so maybe I should sample the boots, maybe, but the boots are walking around quite a lot. So, and the boots will carry a lot. So maybe I should sample the wall of the habitat. Maybe I should sample the beds. What should I sample in order to make sure that there's that there is or isn't backward contamination? And we decided, because we had a limited amount of equipment and time and resources, we said we can check the gloves, because before and after EVA, in this specific scenario of the DMAR simulation, they use a lot of their gloves and they don't have any rovers. So we said, we should probably check the gloves before and after extravehicular activity, because the gloves are sterile. When they put them on inside the habitat, they would be sterile, and when they come back, we would get a glimpse of what might be happening on the gloves and what they might be carrying back. That would be the best resolution we can get. And so if you look at does it say here? Right. So basically before, like I said, it's before leaving the habitat and after, upon returning from the EVA, we would sample those gloves and we would check again through DNA profiling what kind of microorganisms are there and whether they're different from before and after. And what we found out is that we found that the before clustered differently than the after, sorry. So as you can see, the before clusters, okay, before they went into EVA, they clustered together. These are like, semi-sterile gloves that you go and you put on, and then we sort of like used a cotton bud in order to make sure to swab them and make DNA extraction out of that. And then we had a similar population composition before, but after samples are like all over the place. They're very different from the control before, but we have very different results depending on the samples and on the day, and do not cluster with controls. So here, each column is, if you can see below, it says before and after, and you can see there's a bit of a difference between them. And what we see is that's sort of an evidence of a backward contamination that happens on the gloves of the analog astronaut in a DMAR simulation. When we look at backward contamination, what we're looking for basically is stuff that we found in the soil samples, microorganisms that we found in the soil samples, and then microorganisms that we found on the gloves of the astronauts when they came back from their EVA. And what we find is that there is 44 shared bacterial families. And that means that in this specific setup of a DMAR simulation with that specific spacesuits and how they were treating everything and where they hang those spacesuits and how they come back and forth, then you had 44 shared bacterial families. Doesn't mean anything about a real Mars planetary, field expedition, but it does give you a sense of how simple it is to test for these things and what does it take in order to monitor if we do have or not. This is just more specific samples about chloroflexia and Solirobacter. Chloroflexia, again, it's a photosynthetic green non-sulfur bacteria, and how that was possible that we found it back as a cross backward, which means basically C is a psychrobacter that we find near the sanitary, and then we found it all of a sudden in one of the soils. So maybe we had like a cross backward contamination, but we're not certain, and that requires further studies. To summarize what we did in this analog mission and what can be done in a field expedition on Mars is that we looked at forward contamination was detected in the air, which is most adjacent to the habitat. And what I want you to keep in mind is that this simulation did not have any rovers or any buggies or anything that would spread even further and faster all the microorganism population that the human came with. So they were just walking around pretty much. We had a possible backward contamination with 44 species with environmental bacteria carried on the astronaut gloves. And I just want to emphasize that we didn't implement any special sterilization protocols when the astronauts came back, when the analog astronauts came back from their missions. We did not do any type of special sterilization protocols for the gloves, the boots, or the spacesuits. And when we're talking about Psychrobacter, which is a species that some of the strains are associated with pathogenic behavior in humans, but when we found it, it's due to forward contamination. It wasn't there to begin with. It came with the crew, and the crew was in good health, so we didn't find, of course, any health issues with that. But its return to the habitat is an evidence of backward contamination because we found it on the gloves as well. And what we were able to prove with this specific research and this specific simulation is that you can find these missions as an established setting to study contamination questions. And there are many more contamination questions. For instance, do the microorganisms mutate over time? Do they get more pathogenic? Do they become less pathogenic? There are many other questions that you can ask, and then you can test for them while you're in the field doing an analog mission or space simulation. And the ability to control and reduce forward and backward contamination is crucial for the future of humankind in space. Those are big words, but basically you can find much more detail in the published article, which is Temporal and Spatial Analysis of Forward and Backward Microbial Contamination in a Mars Analog Mission in Frontiers in Astronomy and Space Sciences. And we would like to, aside from sterilization technique, we would like to suggest this is a way also to monitor the situation using advanced molecular biology methodologies in order to check what's happening with the microorganisms within the habitat, around the habitat, and also further away. So these are all the different organizations which helped me a lot with this. From the Weizmann Institute, this was led especially from Tel Aviv University and the Weizmann Institute of Science, with help from the Technion and the Eco Encounter Study Institute. It was supported by the Israel Space Agency, and and we would like to thank you, and the Minerva Center for Life Under Extreme Planetary Conditions. And I would like to open the floor for questions and let you know that I'm going on an Antarctic expedition next year, and I looking forward. You're welcome to look at the website and also explore possibilities of cooperation. So thank you very much.
Thank you, Dr. Abramovich. Our next speaker is here, and we are running about 4 minutes over time. So students, if you have any questions, please put them in the chat. And Dr. Abramovich, if you could just stick around for a couple of minutes, if you can, that would be
Yes,
great.
I can. No problem. I'll
I'm
stop my share
surprised.
now.
Actually, we usually have about 10 questions by now, so I don't know if people are just tired.
Yeah.
It's been a long couple of weeks. They've learned so much. And I would like to introduce, thank you, Dr. Abramovich. Wonderful, beautiful talk.
Thank
Um,
you.
I would like to introduce Hiro Ono, Dr. Hiro Ono, a research technologist and group supervisor of the Robotic Surface Mobility Group. His primary interest is to enable the exploration of previously inaccessible planetary destinations through innovative robotic systems, combining versatile mobility and intelligent onboard autonomy. And his title is Life on Mars, which I love, and that's perfect right after the beautiful talk by Dr. Abramovich. Welcome, Dr.
Yeah, thank you. Of course, it is inspired by that song, right? I guess you know what
Right.
I'm talking about. Good to see you guys. I suppose most of the audience are is students, right?
Yes. Yes.
Okay. Which grade are they typically?
Ages 13 to 19, so middle
13
school
to
to high school.
19. Okay. Oh, nice, nice. And I suppose everyone is a space geek, right?
I think so.
Okay. Cool. My name is Hiro Ono. I am a planetary roboticist and technologist at the NASA's Jet Propulsion Laboratory in Los Angeles, California. Well, to be precise, Pasadena. Anyways, I've been working on the Mars rover Perseverance mission, among many other things. So that's what I'm gonna mainly talk about. But I gonna also talk a little bit at the end about the exploration beyond Mars. Okay, so let's start. Uh, I believe I have 30 minutes, right? So I will make
Yes.
sure that I will stop talking at least within 8 hours.
Okay. Cut you off at 7.5.
Okay. So do you see? Oh, okay. I have to share the sound actually. Um, excuse me. Hopefully things good. Share. All right. Do you see my slides?
Yes.
All right. So, we've been exploring space, right, since 1957. And of course, we have a question. Why do we explore space, right? And the answer is not singular, of course. Everyone has different answers, but to me, it is to answer some of the most important and profound questions, not just for science, but for humanity. One of the most intriguing questions, at least to me, is are we alone in the universe? Because we, life on Earth, are in 4 billion years of solitude, right? We don't know if this universe is full of life and we just haven't met others yet. Or maybe the universe is just a lifeless extent of space and time with only one pillable dot here on Earth, right? We just don't know where we came from. We don't even know what the definition of life is, right? And we want to know. And to me, that's one of the most compelling reasons to explore space. So, excuse me, let's talk about Mars specifically. Right, Mars, as of now, is not a good place to live for us, I suppose. If you look at the pictures from Mars rovers, it doesn't look like very, hostile environment. It is actually, right? Temperature could be like, I think average temperature is minus 60 C. Atmospheric pressure is just 1% of the Earth, and it's composed mostly with CO2, and the surface is covered by very toxic uh, um, materials. So that doesn't promising look look promising, but actually the previous NASA's missions to Mars found multiple lines of mineralogical and geomorphological evidences that water existed on Mars about 4 billion years ago. So, most likely, Mars was a planet of water like Earth, right? In the ancient past, it's about 4 billion years ago. Now, this timing, 4 billion years ago, is very interesting because around that time, something interesting popped up on Earth, right? Do you guys know what it is? Anyone?
Life.
Anyways,
Life.
Yes, life, right? We don't know why, we don't know how, but somehow life popped up on Earth around 4 billion years ago, right? And that time, right next to Earth, there was another planet that looked quite similar to Earth, right? So it's not probably a wild idea to think that life might have popped up on Mars around that time too, right? So that's the mission of Mars 2020 Perseverance rover, right? Um, so Mars might look like this, right? Planet of ocean. So, right, is there what was there, right? Life. Maybe it's not the green monster like that, but maybe it's microbes, right? So, can we find an evidence? Or evidence of it's hard to prove the inexistence. So what we did was to send the rover to the place that we think most likely that life was there if it existed, right? So, anyways, that's our rover mission, and that's what I have been worked on for since more than 10 years ago, right? Long time. Anyways, so it was
and smoke
launched
on its way to the red planet.
in
And
2020,
liftoff.
already 5 years
As the
ago.
countdown to
That was
Mars
in the
continues,
middle of the COVID
perseverance of
pandemic,
humanity
so
launching the next generation
I
of
wasn't
robotic
be able
explorers
to
to the
go
red
down
planet.
there to see the launch, unfortunately.
And smoke.
So how did it arrive on Mars? That was quite interesting, actually. It used a very dynamic way to land on Mars. Um, Here's a movie. So the spacecraft took like 6 to 7 months to get to Mars. So that was on February 18, 2021. So the rover was put in a capsule and that plunged into the atmosphere of Mars directly from the interplanetary trajectory. And the first thing it did was to jetson the cruise module that is not needed for landing. And since it has atmosphere, right, the spacecraft decelerates, reduce the speed very quickly with by the friction with atmosphere. So this capsule goes down the sky of Mars like a meteorite, and it actually, is hard and easy, right? Hard because we have to deal with this heat, but easy because, we do not need to decelerate using rocket. So once the atmosphere, we deploy the parachute, and that further reduces the speed. But remember, Mars atmosphere is only 1% density of the Earth. So only the parachute is not enough. That's why we separate the rover in the sky and use the sky crane to reduce the speed to zero, right? So this sky crane flies with rocket engine. It does two things: kills the relative velocity and also do onboard image processing to avoid dangerous lifestyle automatically. And it hovers about 20 meters above and lower the rover. Cut the tether. The sky crane is fly away and rover land on the ground, right? So that's how we landed on Mars. Okay, so that was the computer graphics. Here's the real footage at the landing. So
Tango
sky
Delta.
crane, top
Touchdown
left,
confirmed.
rover,
Perseverance,
down, and here is how I
ready
celebrated.
to begin seeking the sky.
Again, that was in the middle of the COVID, so I celebrated home with my family, with my 4-year-old daughter. Look at how messy our living room is with 4-year-old kid. Little trivia: this is the parachute. Actually, took this picture was taken on Mars during the descent using the upward camera. Now, parachute has some weird pattern, right? Do you know what it is? So this is an ASCII code with their mighty things. That's the motto of JPL. So where did we land? We landed on the Jezero crater on Mars. So this crater, about like 50 kilometers across, was once a lake, right? Huge lake on Mars, and two rivers were flowing into the lake. One of them on the west end of the lake um, formed a beautiful delta, right? And this delta was the target for exploration. Why? One, obviously there was water, and two, delta is a convenient target because, the river for billions ago collected all the materials from upstream and dumped everything in one place, right? And also, using the orbital observations, this color is not a true color. This is a false color, colored by the types of minerals that they found from orbit. And one thing that particularly made scientists excited is the carbonated minerals, right? I don't know the detailed story, but somehow they think that carbonated rocks have good chance of preserving the evidence of life. Okay, so the rover landed here, a kilometer or two away from the delta. So this is the picture, panoramic picture taken from first glance. Beautiful, right? So the mountain at the back, that is the rim of the crater. This rim, there's a point that the rim is lowered. This is the place where a river flew into the crater, the lake, and there's a hill, right? In front of the crater rim. This is the delta. So, the rover landed at the lake floor, ancient lake bed, and then drove up to this hill, delta, climbed up, and then again climbed up the hill, the rim, to get out of the crater. All right, so a little trivia. The rover is actually very slow compared to the cars that you are familiar with, right? So which one is the closest to the speed of Perseverance? How slow it is? Snail slow, ant slow, sloth slow, or human baby? That's actually my daughter when she was like half-year old. Um, you can probably type your guess in the chat. Can you see the chat somewhere?
They're coming in. They're coming in. Yeah.
Okay. All right. D. All right. One vote for D. Anyone else? A. C. Okay. I wonder what prize you will get if you get right. Probably none. D. Okay. Answer this. So the answer is C. Sloth. 0.15 miles per hour, or about no, miles per hour. No, I get it wrong. This is a kilometers per hour. It's about 0.1 miles per hour. Anyways, sloth is slower than ant, which is interesting, right? But anyways, so rover is a sloth. So this here is the movie that I took at the Maziar at JPL. This is the real speed of Perseverance, right? Very, very slow. But with this speed, so far the rover drove about 35 kilometers. As of March, so I think this is now it is like 37 or 8 kilometers. I don't know the exact number. So as I said, it drove at the crater floor, climbed up to the delta, and then went we climbed up the crater rim. Now we got outside of the crater. So my job, I mean, I did a few jobs. My main job on this mission was to develop the autonomous driving software algorithm for the rover. So actually, and this is the major difference from previous rovers, the majority of the distance, about 90% of this distance, was driven autonomously. Now, here's a trivia. Now, many companies, Waymo and Tesla, are working on autonomous driving on Earth. We've been driving on Mars autonomously since 2004, I believe. So, autonomous driving on Mars preceded the autonomous driving on Earth. All right, I'm gonna be a little bit rush. So there are 23 cameras on the rover. There are many, right? All right, another trivia. So the rover has some superpowers, right? Which one do you think? A, shoot lasers from eye. B, X-ray vision. C, create oxygen. D, take selfies. I don't, maybe taking selfies is not a superpower particularly, but which one do you think? C, D. All right. Yeah, of course you can take selfies, maybe, right? I don't know. All. Okay, the answer is actually yes, Nicole, you're right. It's I didn't say that the answer is singular, right? So it can shoot a laser. Why shoot a laser? It's not exactly to blow up rocks. Well, it does a little bit. So it melts the rock a little bit, and it observes the spectrum coming back from the vapor to figure out the composition of the rock. It has a X-ray vision. It has the ground penetrating radar so that while driving, it can see underground, right? It creates oxygen. We have an experiment, we have an instrument called MOXIE that takes CO2 from the atmosphere and creates oxygen. Why? Obviously, to get prepared for a future human spaceflight, right? Robot doesn't need oxygen, human does. Taking selfie, of course. We have a camera. One of the 23 cameras are at the end of the robot arm. So we use that to take selfies. Here's one of them. Now, this is Perseverance. Perseverance has a friend, Ingenuity, right? That's a helicopter that the Perseverance took with her to Mars. And Ingenuity became the first powered aircraft that flew successfully anywhere other than Earth. So Ingenuity is basically the Wright Flyer on Mars. And in fact, we put a piece of a fabric from the Wright Flyer on the back of the solar cell of Ingenuity. Now, another thing that Ingenuity is doing is to drilling the rocks, right? So why? To collect the samples, right? Why collecting samples? Because we want to bring them back to Earth. So here is how it would work. So, hopefully another spacecraft, this is Perseverance, right? Perseverance rover uses this drill to core the rocks and take samples. And then another spacecraft gonna land on Mars, hopefully someday, right? And the rover gonna drive up to the lander and transfer the sample tubes to the lander. Now, this lander has a rocket to be launched from the surface of Mars, right? Now, the way to launch the rocket is interesting. It throws the rocket to the air and ignite in the sky. Right? If you want to know why, I can answer you right now. Right? And that brings the samples to orbital Mars. Then there will be another spacecraft that's gonna catch the capsule on the orbit. And will come back to Earth. Exciting, right? So why we are bringing back samples, right? Finding the evidence of life is not easy. We need many instruments to analyze the rocks. Best way to do so is to bring them back and it's all the interesting instruments that we have on Earth to analyze. Now, do we think we're gonna find the evidence of life? We don't know, but we did find interesting rocks, right? For example, the rover found what we called leopard spots on the rock called Cheyava Falls. And again, we don't know, but there are many hints that excited scientists a lot. For example, we used our onboard instrument, Sherlock, and we detected organic compounds and leopard spots. Typically, we saw these kind of things on Earth. It typically indicates interesting chemical reactions that could be used by life as an energy source. And also has a clear evidence, this olivine rock piece, that water was once there, right? So we have all the interesting conditions, energy and water. So we'll see if we're gonna find something. A little trivia on Mars. The sunset is blue. I would love to see it one day. All right, I will spend 1 minute to talk beyond Mars, right? We're talking about water. Water is everywhere in the outer solar system. And in fact, there are many moons of Jupiter, Saturn, and beyond made of ice, right? One very interesting moon of Saturn called Enceladus has a geysers, right? So why it has geysers? Because this little moon, about 3 times bigger than the Death Star in Star Wars, right, has global subsurface ocean. There's an ocean below the ice, right? And there are 4 gigantic crevasses through which the water vapor ejects to space, forming a ring of Saturn. Interesting, right? Is there life in there? Right? That's a question. Or maybe just, microbes or maybe fish. So we imagine a future mission like this. We're going to land on Enceladus. It looks like a snake, maybe. Crawl on the surface, get into the crevasse, and dives down, goes down to get to the liquid water ocean. And white snake, it's because, we don't know how narrow or wide the crevasse is, right? Once we get to the ocean, we sample the water, look for life. So that's our concept. That's going to take many decades, but we prototyped the robot and brought it to Canada, a glacier in Canada, to test it. So we tested the mobility on the surface and used many gates, locomotion patterns to move around on difficult terrains on ice. Also, the glacier has crevasses, right? So we used some of those to test the vertical mobility. This robot, this is version 2, looks more like a spider, but same as the snake robot's mechanism. So this robot is hung from the top, put in place in the crevasse, and then it expanded the arms to push the walls against each other, and then use the wheels on the side to move vertically. I believe we are the first to achieve the vertical mobility on natural vertical shafts on glacier. So I thought we came a step closer to our ultimate dream, and this is how happy we were. All right, so I hope you guys are excited. I want to present just one more slide, not as a GAPO employee, but as a private citizen. Maybe you know the news um, that in the presidential budget request, 47% cut uh, of NASA's science budget is proposed. What it means, many ongoing and upcoming missions will be canceled if this budget cut materializes, right? Including the Mars sample return that I just talked, two Mars orbiters that are already there and working to support the rover's data relay and also collecting science, Maven and Mars Odyssey, as well as many planetary missions. Juno, the Jupiter, New Horizons, now in the interplanetary space. No, not interplanetary space, but moving beyond the Neptune orbit. Nancy Grace Roman telescope as well. Two Venus missions and Osiris Apex. Now, again, this is my personal opinion, not the opinion of NASA or JPL at all, but this gonna be a substantial loss, not just for science, but loss for humanity. So, if you think that exploration of space for science is valuable and something that we need, please help us by raising your voice, right? America is a democracy, right? It's not like any single person can decide. So if many people can raise their voices, things could change. I will stop here. I wrote my super long thoughts, opinion in my blog, so please take a look if you're interested. Okay, so I hope you're excited. I will stop here. Thank you very much.
Thank you, Dr. Ono. We have a question. Won't the carrier holding the samples explode due to aerodynamic fragmentation?
The aerodynamic, sorry, the carrier of the samples.
Right, the Mars sample return.
No, I think the question is regarding to the launch of the sample carrier, I suppose. Um, so, well, of course, there's going to be atmospheric pressure that the rocket has, but it is designed to, when you launch a rocket, you have to go through what's called max Q, the maximum aerodynamic forces, and rocket or anything is designed to be able to withstand any aerodynamic pressure with a lot of margin. So, no, it should be okay. Of course, it's not an easy engineering problem, but there's a solution.
Okay, and there was a question for Dr. Abramovich also. Do you think the backward contamination is possible from a human Mars mission? And if so, what technologies can we use to prevent it?
Yeah, great question. And actually, I was the last portion of your talk. Yes, I am concerned. Um, let's see. I wanna, speak carefully. I think I don't have to talk about the backward contaminations. So, an impact. As for the Mars sample return that NASA is thinking about, we are super cautious, right? We're gonna put the samples on the biosafety level 4 facility it's a level of the safety required for handling super dangerous viruses like Ebola or something else, right? So uh, now when humans go to Mars, all the planetary protection story is gonna change because we human body has it's full of microbes, and so for one thing, Mars might still have very precious ecosystem over there, and we human beings have made many mistakes on Earth of destroying precious ecosystems. We should not repeat, we do not go to space to repeat the same mistakes that we did on Earth, right? We go to space to be wiser, which means that I strongly think we should not just go there without caution and destroy, which does not mean that we should not ever send humans to Mars. What we need is just a careful investigations. We need to investigate first if there's life on Mars. If there is, how we can, mitigate any negative impact on them or any negative impact of backward contamination. We can learn a lot through sample return. So, you know, a little bit of criticism to what Elon Musk is saying. Again, my own opinion. It's not just, hey, let's build a huge rocket and send humans to Mars. It's not that simple. It has to take steps. We have to learn a lot about Mars, about life over there, and then we will have a safe and reliable way to go to Mars. That's my opinion.
Okay, thank you. And I'd like to give Dr. Abramovich a moment to answer as well.
Thank you very much. Yeah, I totally agree with Masahiro, but we really need to define the exact backward contamination scenario you were thinking about when you were asking the question. So there's backward contamination because you got sample back to Earth, and there's backward contamination because you had a crew on Mars, they went out to survey the environment and they came back. Currently, I'm not worried about backward contamination from Mars. I think it's a very low risk issue. The problem I have is that we have so much sensitive equipment that we are using in the biological sciences that we will encounter very quickly the problem of, are we identifying something that was on the Martian surface or subsurface, or are we identifying something that came with us? So basically, even before we set one foot or we do one analysis on the Martian soil sample from a biological perspective, we're going to have like a major database and a long list of all the stuff that we brought with us already, that we sampled with us, and that we did the analysis with us in order to subtract it, okay, to remove it from any other results that we're going to get while we're doing the analysis on the Martian soil or the Martian subsurface or Martian cave walls, for instance. So we cannot sterilize, but I say we can monitor, and of course we can be very careful and develop those protocols here on Earth and also on the ISS. One of the things that the ISS could be, it could be a hub for getting all these sample returns. Let's say we now have a flourishing 2 decades of sample returns from Venus and from the moon and from Mars and from asteroids. Instead of putting them back on Earth, we can use the ISS as a certain area, as a buffer area, in order to check those things first, because we have quite a lot of equipment and we have good people on the ISS that can test these things. And then we can say, okay, let's put it back on Earth. That's my own opinion. Thank you.
Thank you so much to Dr. Ono and Dr. Abramovich. Thank you, students. I will be in contact. If you have not sent your summary, your outline, please send it. I will be checking in with you. Please watch your email. I will be sending updates and a mentor to you within the next few days. Thank you to our speakers, and I will see you, students, and talk to you in the next couple of weeks. Thank you so
Thank
much.
you, everybody, for your time and for the invitation. It's been fantastic for me. Thank you so much.
Thank you. Stay safe.
Thank you,
Yes, thank you.
guys.
Thank you.
Ciao,
Bye.
ciao.
Bye.